Okay. Good evening, everyone. My name is Jongyoon Han from MIT Electrical and Bioengineering. Today, I'm going to talk about some of the microfluidic effort on separating rare cells from various kinds of biofluid. So there has been a very exciting development in the area called microfluidic, which is the new concept of inertial microfluidics. And this is very innovative because it combines the microscopic control that enables the very good separation with the macroscopic volume throughput. And I think Professor Toner's group at Harvard and many others actually pioneered this area and still in the early stages of development. We have been working towards applying some of these innovative concepts for sample preparation of biofluid, especially for separating background cells from various kinds of biofluid, including blood and others, and perhaps collect the rare cells. And this is not a new concept, and many groups are working in this. And we have had many successes, but still there are a number of challenges remaining. Some of those are really obvious. First of all, the blood, for example, is very complicated objects. So you have a 40% volume basically filled up by red blood cells. So it's a very high background content. And whenever you try to separate these 40% hematocrit sample, you run into a problem. And then even these inertial microfluidic system doesn't really work for such a high hematocrit samples. And because of that, you have to dilute the blood, and then that limits the overall throughput. So, in order to address some of these challenges, we have been working in a new way of separating cells from the blood, but still allowing high hematocrit samples. So by that, I mean 20 to 40% hematocrit samples. So it wouldn't require you to dilute the sample blood that much so that you can actually increase the throughput. So the details of the idea is probably beyond the scope of this talk, but it is suffice to say that it utilizes the very simple ideas of spiral microfluidic channel where the blood or slightly diluted blood is injected into the channel with a very high speed. And because of the delicate interaction between the fluidic channels and the cell, all these background cells, such as red blood cells and white blood cells, in this particular case, can be segregated into one side of the channel in this outlet, while the target cells, in this particular case, the CTCs in the blood, can be focused in the one other side. So, again, this is not the first device that can separate the CTCs out of the blood. What I'm trying to say is some of the salient features of this particular channel, meaning that it just works even with the high background concentration of the red blood cells. So in this case, we are showing that we can actually work with up to 20 to 40% hematocrit sample, which is almost like a raw blood. And if you run this device twice or third time, we can enrich the very low abundance cells like CTCs while rejecting all these other background cells. For example, we are achieving almost 9 orders of magnitude rejection ratio for the RBCs. That's practically removing almost all of the RBCs from the given input blood sample. The other good thing about this device is that the device doesn't really touch the target cells. What that means is that everything happens in solution. At the end of the device, we have a tubing that collects the solution that only contains these target cells, in this case, CTCs. What that means is that we are, capable of collecting some of the other targets, such as tumor emboli or tumor aggregations. So that's maybe interesting for the downstream analysis, because now we can actually have a sample preparation device that can collect some of the low abundance species. So showing some of the early results from our group. So we applied this for collecting the CTCs from the patient blood, and the workflow is very simple. You take the whole blood, you dilute it probably twice, and push it into this simple microchannel, and we collect the cells contained in a large volume of solution, and we can do whatever we want to do. We can put them on the 96 well plate, we can try to culture them, in whatever matrices or whatever conditions. So, one point that I want to make is that this is really a sample preparation devices that can be used to collect the rare cells. And the recovery is the strength of this technique. So some of the earlier work is really a preclinical work done in Singapore with the collaborators there. We were able to collect, patient CTCs and compare that with the healthy patient. And, there's a very limited number of patients, but still we are getting a good correlation. And then at the downstream, what we do is exactly the same as other standard technique. We just plate them on the plate and we do the standard antibody-based staining, and we can count the CTCs. And the throughput is relatively high. A single channel can process up to 3 milliliter per hour per channel. And if you can stack multiple channels, we can achieve 10 mil per hour. So it's pretty fast. And also, if we can choose to lyse the RBCs, then we can actually process the larger amount of blood in a much shorter amount of time. In fact, we were able to process up to 7 mil of blood in 10 minutes just to collect the CTCs with the lysis method. So it's very exciting. It's a very high throughput, so we can actually handle many types of biofluid. Now, I'd like to show another examples of using these types of device, because this device is really a nice sample preparation for any kinds of biofluid. In this case, we are taking bone marrow aspirate or maybe a mesenchymal stem cell sample, which is known to be very complicated and diverse. And then what we are trying to achieve is that, starting from large volumes of such fluid, and we try to select the cells that are smaller in size, and there has been a number of publications suggesting that maybe those smaller MSCs are indeed stanier in the sense that they have higher differentiation potential. So this is one of those experimental results. We can actually size separate these diverse MSC fractions into two different populations. So D high is the larger MSCs, and D low is the smaller MSCs. And if you can see them, the outlet 1 collects the larger cells, outlet 4 collects the smaller cells, and if you grow them in the plate, you can see that morphology is clearly different. These are really spindly kind of morphology, as they call it. And what that really means is that they are really different cells, okay? So these D high cells versus D low cells have a clearly different differentiation potential. So now we can actually see whether D high cells has a potential to differentiate into, fats or other, and they do not, and they can only differentiate into bones and a few other targets. But then these smaller cells tend to have a higher differentiation potential. So this is a very preliminary result, and we are just about to send this out. But the point that I want to make is that what we have here is a relatively simple device. So this is a device, it's a piece of PDMS, and all we need is a syringe pump and then push the cell solution, and we collect the cell in the other tube. But with that, we can actually process many different types of biofluid, including blood and bone marrow aspirate, and potentially peritoneal fluid and others. And most of these biofluid preparation, usually the problem is that the high content of RBCs needs to be removed, and that's exactly what we can do the best. And last piece of information is that although this is a fast flow microfluidic system, we have confirmed that this is very benign to even the sensitive cells, such as neutrophils in this case, and we were able to culture the mesenchymal stem cells out of these spiral channel. So that's it for today's presentation. Thank you for listening.